watch me bring the fire, set the night light. Shoes on, get up in the morning, cup of milk, let's rock and roll. Kink out, kick the drum, rolling on like a rolling stone. Sing song when I'm walking home, jump up to the top of the brown. Ding dong, call me on my phone, nice tea and I'll get my ping pong. This is getting heavy, can't hear the bass, boom, I'm ready. Life is sweet as honey, yeah, this beat chain like money.
Now, the new chapter of chapter 2 deals mainly with the evolution, starting from the origin as scientists presently think how they have been unfolded to the actual several aspects of the human evolution, the old way through that we are going to take a look at. So it actually takes two different part of the aspect of the evolution. One is the very small scale, we call it the chemical evolution, and the other one is the more familiar Darwinian evolution, as we have briefly seen in the previous chapter. But the way I'm going to go over this chapter, unlike the previous semesters, I'm going to reduce the amount of time that I get to spend on, although this is actually very extensive material, but I'm going to skip several of them, in particular like the creationism versus evolution aspect. I'm going to almost omit those discussions for this semester for some reason. So let's take a look at to explain the origin of life. There has been two alternative ways, and one is spontaneous and the other one is called biogenesis. Maybe when you think of the spontaneous generation of life, you may actually think a natural way of generation, but actually quite the contrary. So this before the emergence of a biogenesis theory, actually the spontaneous generation theory of life ruled the entire the philosophical and scientific community, mainly due to the religious influence on the Western culture of Christianity. So here it states that this living life forms can be generated spontaneously if the proper conditions are met. But these proper conditions, what they mean by these proper conditions is divine interventions. Which means God's creation. So this has been the predominant idea of the Western culture due to, like I said, the Christianity influence, until modern science disprove that is not the case. On the other hand, this biogenesis maintain that life can only arise from the pre-existing one, which is the case around us in these days. And the one famous figure that ultimately proved that this biogenesis was a correct idea was Louis Pasteur, was a renowned medical doctor and microbiologist who first time invented the vaccinations by inventing this new experimental apparatus, which unlike a previous semester, I won't go over in too much details about this experiment and the setup. The only thing I want to stress over here is he using this strange shaped flask, he created a contamination free environment. So as long as he maintained this contamination free environment, then under this contamination free environment, no life, no spontaneous life emerged. So in other words, the previous thought spontaneous generation was all due to the contamination of a microorganism from airborne microorganism. Well, in these modern days, that idea was just a childish ideas, but that's some time ago when the level of understanding in science was not really as much as these days. So that was it. However, once recognize this biogenesis as a means of new life form generation, then immediately some paradox happens. Then how did the first life form, if all life forms must come from pre-existing one, then how come, then what about the very first life form is the problem. So to overcome such paradox, so scientists came up with another paradox. So in this biogenesis theory, the very first one must be generated. It's inevitable. This very first one should have been generated spontaneously. And for some reason, all of a sudden, no more spontaneous. Even though I became existing, my existence was made possible by spontaneous generation, but no more such trick. So from now on, all this new generation of life must come through me is the idea. Believe it or not. So in other words, the very first single event was spontaneous generation, followed by the Darwinian, the biogenesis. So this single event of a spontaneous generation is regarded as the chemical evolution. And from then on, all the new life forms through biogenesis only. And this biogenesis involves the different types or different modes of evolution. We call is the Darwinian evolution, as we have seen in the previous chapter. So this chemical evolution, let's take a look at a little more. So this specialty about this particular spontaneous generation is the new life form was made from non-living matters. As I have briefly shown in the very introduction lecture in the beginning of the semester, the material, all these materials, all these components, chemical components, are non-living matters. Okay, and all these materials somehow collected themselves and somehow got the protective barrier, which is cell wall, cell membrane. And now the other key component here, the genetic material you have, now then we declare this as a new and the first life. And maybe this is the one that we recognize as Luca, the very first ancestor. We call it as the last universal common ancestor. And from then on, so this is obviously is capable of dividing, so reproducing itself thanks to the presence of this, the genetic material. It's the whole idea about this chemical evolution. So this whatever this coacervate idea, which is obviously of course incorrect, but the only value about this idea of so-called coacervate, which was proposed by Oparin some time ago in the early 20th century, is a self aggregation of material is possible and actually can be regenerated in the laboratory setting in these days. So in some cases, some organic material can clump together, and then they can form some living cell-like structure. The only thing missing over here is what? The protective cell membrane over here. But anyway, the only value of this coacervate idea is it is possible to have this clumping together spontaneously without any some kind of intervention, without providing some artificial energy input. Because if you think about it, why this is something that is special is this one is against from scattered individual materials to have this collection of aggregation, which is moving from higher entropy to the lower entropy, which is not going to happen spontaneously. But in reality, it can, for some specific reason. It is not actually violating any of the rules of physics, like the second law of thermodynamics. With that, actually, the scenario of such a spontaneous generation through chemical evolution goes like this, as scientists envision. So first, you have to be able to synthesize this organic material, because organic material, as we went over, can only be found in the living cell. Okay, that's why it earned its name organic. So these organic monomers, just like should I go over this organic some of the, yeah, I will at least go over some organic macromolecules, 4 different types of organic macromolecules. Anyway, so here let's actually distinguish only the difference between monomer versus polymer. And polymer. So monomers are, as you can see, literally individual molecule. Yeah, separated, isolated molecule. Polymers are combined. So all these monomers are put together, and then now you have this polymer. Get it? Now, to assemble polymer starting from monomer, you can easily understand that it will take the energy, right? Because this direction, the entropy, is a decreased entropy wise, because it's a synthetic. So is this catabolism or anabolism? This is an anabolic reaction, right? So somebody has to provide the energy here, but that somebody, who can be that somebody, is the issue, because no one was there except depending on who you are, except the that the creator God. So that's the idea. So how can it be possible? And besides, this is not a simple any other type of molecule, but this is an organic molecule that carbon containing. So this organic monomers, carbon containing molecule, organic monomers. The examples of organic monomers. What are some of the examples of organic monomers? Relatively simple molecule, but containing carbon. Is such an example is 1 glucose. Get it? Yes. And like other type of simple sugar, glucose, and what else, and fructose, and several different types of simple sugars containing only small number of carbons. In the case of glucose, it has 6 carbons and having some oxygen and hydrogen combined through specific chemical bond. Another such example of organic monomer is fatty acid. We all know, hopefully, what the fatty acids are. And another is amino acid. Yes. And another example is nucleotide. Remember? What did I say about nucleotide? Nucleotide, 4 different kinds of nucleotide assembled together make DNA. So DNA is a polymer. So in other words, all these different organic monomers are the base, the components of building respective polymer. So this glucose now later is the basic unit of building more complex sugars like, for example, starch or glycogen. These are all sugars, but more complex sugars. So like starch, as we all know, they are plenty abundant in our food, in the, like a meal, the rice and the bread, and glycogen is another such complex carbohydrate made in the animals and stored. Although whereas a starch is the one corresponding such complex sugars produced and stored by plants. Fatty acid is another such organic monomer from which the more complex is now because of fat and oils. So those two more complex lipids synthesized from this fatty acid. Amino acid, now this is the building block of generating protein. And nucleotide is like DNA, and the cousins of DNA is RNA, all together called nucleic acid. Are their respective relationships. So being able to synthesize spontaneously at first these different types of organic monomer is the first step towards building more complex molecules. These are the examples of macromolecules, right? Which are the absolutely necessary materials to build the living cell. So now then, starting from organic monomers, like for example, starting from glucose, you should be able to, without any intervention of anybody else, should be able to spontaneously synthesize something like glycogen or starch or similar but quite distinct in chemical property in cellulose. So this cellulose serves as a building block for the plant cell wall, the hard material on wood. So this, can it be possible? This abiotic means spontaneous synthesis. Because why do we concern this spontaneous synthesis of these polymers? Because this is apparently against the nature, the law of the nature, like second law of thermodynamics would predict that is not possible to happen spontaneously, because somebody has to provide the energy over here in a specific way. Now, these organic polymers, now as I said, another name is organic macromolecules. Then using this, now the spontaneous assembly of a cell. So in other words, all these materials should be present. Like first you have to be able to build cell membrane. Obviously cell membrane is built with the lipid, some specific type of lipids, complex lipid. We call it as phospholipid, or we will see that chemistry a little bit later on in this chapter. And inside you should be able to create some all different stuffs, including, if you were like, especially if you were an eukaryotic cell, you should be able to build some organelles. In order to build organelle, at least you should be able to build a barrier, which is membrane based, lipid based. All membranes are lipid based, like starting from fatty acid. This is very similar to fatty acid, but not exactly the same fatty acid that I mentioned. And also inside of the nucleus you should build this, your chromosome, the DNA, which is basically built from the nucleotide. So all these organic polymers should be there to have some kind of a specific organization, now we call as a cell. The reason over here is a pre-cell, and they think, at least for the prototypical first type of cell, is not as complete or as perfect as these days the cells as we see. But at least in order to reach that point, you should be able to have some kind of this magic involved. And once again, and another very important thing is, at least you have to ensure that the presence of this self-replicating molecule, your genetic material. The reason why this DNA can serve as a genetic material is its special feature of being able to go through this replication. What did I say about the very important issue before going on the cell division? You should duplicate your whole genome. So this replication, DNA replication, has to occur in a very precise way and a very elegant way of what we imagine in this area. So the mode of a self-replication. So this is a self-replicating molecule. then that pre-cell eventually refined itself into the more complete finished products now that can be recognized as the first ever ancestral form of this whole entire life form. Look up, maybe. So this figure illustrates such events starting from very simple inorganic, mainly inorganic components. So here in this CH4, which is methane, although methane contains one carbon, but methane and carbon dioxide, those elements and molecules, are not regarded as organic compound. But they have this more complex, a little bit of this organic compound, and then eventually get to produce the real organic monomers like this, an amino acid and some of the component in building of DNAs and more complex such polymers like protein and DNA, RNA, you name it. And then they have this pre-cell. The important point over here is all this building synthetic procedure happened spontaneously, which means they are all against, because at each step, each respective following step would have decreased, resulting in the further decrease in entropy. And so you may wonder, how can that be possible? There's some issue that we have to consider, and how exactly that might have happened. So here, let's talk about in the living, the components that constitute the living cell. There are 4 different groups of such organic macromolecules. One is carbohydrate, and the other ones are lipid and proteins and nucleic acid. So, carbohydrates usually in the cell, in the living system, the utility of carbohydrates, there are so many different ways carbohydrates are used, but in one, this is an energy, immediate energy source, the food. The reason why those carbohydrates have been used as a really effective food source is because it is very efficient energy provider, so that we can, by breaking down their chemical bond, we can extract the energy stored inside of this carbohydrates energy, and also energy storage. So like glucose is one such example of an immediate energy source, whereas energy storage, the glycogen in the case of an animal, is the storage form of energy. And also structural form of the so like cell wall, plant cell wall, and also fungi cell wall and bacterial cell wall, basically all of them contain some of the different types of carbohydrates. So that in the case of a plant cell wall, the cellulose is different type of carbohydrate polymer. So instead of serving as something that to be digested to provide the energy, but this one is the one that to provide the protection against some mechanical and the second utility of the carbohydrates is some of the carbohydrate is also in different shape. This is a signaling recognition, as we will see later in the chapter 3. So many carbohydrates serve as an energy provider and the protection and mechanical protection, but sometimes there are many other miscellaneous other details of usages also exist for the carbohydrate. And for the lipids, mainly the fats and oils, but also there is a special type of lipid called a phospholipid, which is a basic component of the cell membrane. And other lipids material like stereo hormones and cholesterols. Is another such important lipids. So in the fats and oils mainly used as an energy reservoir. But in the case of this phospholipid, which is a polymer of the phospholipid, and this is a structural. The barrier is their main purpose. And in the case of a cholesterol, is also, you never know actually real purpose of using cholesterol. And this is the main ingredient, very essential component of the cell membrane. And also some other usage of this cholesterol is cholesterol can also be used in producing the hormone, specific type of a hormone we call the steroid hormone. All these are actually made starting from the cholesterol. So these are the mainly, although of course there are some other many different functions of lipid exist in the cell, but these are the one of the main roles of lipids. In the case of proteins, one very obvious function provided by protein is enzymes, right? Enzymes, as I briefly said, is a biological catalyst. So without enzyme, no metabolism is possible virtually in the cell. So enzymes are so important, and so many different, tens of thousands of different types of enzymes do exist. Okay. And not only that, some other structural proteins do also. Receptors. In recognizing some other oral stimuli outside stimulus, receptors are needed. So that's why on the cell, many different receptors on the cell membrane surface do exist, and all of these receptors are made from proteins. And also not only that, a lot of such transporters on the cell surface. So specific materials are only handled by specific transporter proteins, and then that material can go through this particular transporter proteins. And then you may wonder why only proteins are heavily utilized in these such very important tasks in the cell. It's their structure. Proteins, for some reason, can generate so many different unique shapes. And in biology, it is not as simple as as complicated as you think. It's very simple. So that it's simply shape dictates function. Okay. So if one particular type of material can generate so many different shape, and that particular type of material can provide so much functions. Is that much simple. And nucleic acid, which is made from nucleotides. Nucleic acids are unlike so many different types of proteins and so many different types of lipids, and just like so many different types of carbohydrates. In the case of a nucleic acid, there are only two different kinds of nucleic acid exist. One is DNA, and the other one is an RNA. And the difference we are going to take a look at in later in this chapter. And DNA is obviously is a genetic material. And this RNA is just like in terms of a chemical property, it behaves as if it was a cousin of DNA. It is the deliverer of this genetic information. To do what? To build. So ultimately, all this utility actually is something to do with this protein. But instead of directly starting from DNA to make protein, but it has to go through the intermediate form of this RNA to reach in protein. And the actual particular procedure of this generating this protein from DNA, we are going to take a look at in chapter 4. So these are the 4 different types of macromolecules. And another thing is the property of these macromolecules. Here, carbohydrate, lipids, protein, and nucleic acid, all of these materials in living world, all these things have to deal with the water. So just like our entire body is filled with about 70% water, so is the case of any living cell, whether you are cell or whether you are like some macromolecules inside of cells. So you have to learn how to deal with the water, because life began in the water initially. So there are 2 different, no matter how many different types of materials you are, in terms of the relationship with water, you are either water friendly or water hating. Some materials are water friendly. A more professional terms of water friendly is hydrophilic. Water loving. Why some material would love to associate with water? Because it fits with the very chemical property of water, which is water itself is a little bit polar, which means takes some electrical charge. Water molecule, I don't want to go into any chemistry, but just bear with me. So this is bare minimum, and then no one has to actually know on the exam about this chemistry, but without talking about this, at least the basic chemistry, there's no way I can go further. So water itself chemically speaking is like 1 oxygen and 2 hydrogen. And oxygen itself, this guy is very greedy in getting electrons. So draw the solid line between these two. So oxygen is holding one relationship, chemical bond with one hydrogen, and on the other side, this same oxygen is holding that same relationship, the particular type of chemical bond with the hydrogen bond. But these two poor hydrogens, so every different element has its own electrons. Okay, the very outer most barrier of any element, different number of electrons actually are present. But this oxygen is just so powerful and greedy to take any electrons from anybody else, the other different element. So when you set up this particular agreement of a relationship, and these two poor, weak hydrogens, they do not stand any chance. So all those electrons will be taken virtually by the oxygen. So all the electrons usually will be around the oxygen side. So in other words, even though apparently they don't have any kind of discrepancy in the distribution of the electrons, but in reality they do, so that most of the electrons are here near oxygen. That means their own original electrons were taken, so there occurs some discrepancy in the electron distribution, so a partial weak charge are created. So here this point is a slightly negative electrically, because electrons are the one carrying this negative charge, and because these side their electrons were taken, so they have a slightly positive charge over here. So bottom line is so one water molecule is polar molecule. Polar means charged. It's not electrically neutral. So as water molecule is polar, and the material with a similar chemical property will be able to get along very well with this charged molecule. So other charged molecule. So all those materials that can be labeled as hydrophilic are polar molecule. Either really obviously ionized, like a sodium ion, magnesium ion, all these ionized elements are obviously they are polar and charged. But less obviously charged molecules do also behave as hydrophilic molecules. So they will be readily dissolved, or they have no problem in existing surrounded by water. The main property of those hydrophilic molecules. So that goes for usually almost all of the carbohydrates. They are hydrophilic because by definition the carbohydrates contain many number of oxygens. So if there was plenty of oxygen, then that oxygen will cause these troubles, because they will try to take somebody else's electrons and then become polar charged. And proteins are made of 20 different kinds of amino acids. And their individual amino acids, most of them, not all, but most of these amino acids are actually polar amino acid. So this, I shouldn't say most of them, but some at least some amino acids are extremely charged, and some others are like, little less obvious, slightly charged polar amino acid. So anyway, since the protein, a single individual protein is a huge collection of many different amino acids. So usually protein, at least some part of the one single protein, contains this hydrophilic surface, the portion. So generally, most of these proteins, we can safely consider them as hydrophilic. And nucleic acid, they are extremely polar. As we will see later on this chapter, the actual composition of the nucleotides and the way they assemble the nucleic acid polymer, it is extremely polar molecule. So all these three biologically important and major organic macromolecules are all hydrophilic. The problem goes with the lipids, because unlike the other three types of macromolecule, the organic macromolecule, this lipid, by the nature of the basic chemistry, all the lipids are hydrophobic. Which is water hating. This one really hates being surrounded by water, because water is polar, but this lipids is non-polar molecule. The lipid is uncharged, so it will try to repel. Okay. So it will, as much as possible, to try to avoid to have any contact with the surface of water. That's why if you put a drop of oil onto the water, then it is not mixed. Rather than, instead of being mixed, it will stay isolated. So that's why oils and lipids doesn't get mixed with the water, because of this property. So this hydrophobic property actually plays extremely important role in spontaneous generation of life, actually, as we will see later. So now let's take a look at the chemical means of assembling the polymers from monomer. So how would you actually put each different individual, as I have drawn briefly in the earlier slide, each individual monomers. So if you want to build the assembly of each individual monomer, you have to apply some type of specific glue. Yeah, this glue, in chemical version of this glue, we call as a chemical bond. So for example, you either put some of the bond over here, is the one way to create such polymer. So these chemical bonds are the one making possible to create some polymers from monomers. And there are, so the molecules like element, two molecule. This is another example of the chemical bond. If you want to put more than two different elements together to form a single molecule, you have to use this magic of some chemical bond formation. So there are several different types of chemical bond that exist. So I will have to introduce some property of such main chemical bond over here. So one is covalent bond. Usually this covalent bond, which is the strongest in chemicals, the strengths of the bond, we represent this covalent bond by drawing a solid line, like oxygen and hydrogen. There is a solid line. That means this particular one is covalent bond, and another solid line, hydrogen, like that, and carbon, between carbon and oxygen, two solid lines in each side, which means two such covalent bonds have been established between a carbon and an oxygen. So the reason why this particular chemical bond is called covalent is between this, the mechanism by which this chemical covalent bond was formed. Behind, underneath of this exists this sharing agreement. Between the two parties. So I don't have to go into that details. I would like to avoid as much as possible, so I will just stop over here. So chemical nature of this covalent bond is the reason why they stick together through the covalent bond is they are sharing each part of the electrons together, just as if their own, and then by which some very stable, solid relationship maintains. That's why it is called the covalent bond. And anyway, this is very strong. So, like for example, glucose, chemical formula of the glucose is 6 carbons and 12 hydrogen and 6 oxygens, but these are all connected through the covalent bond. Okay, and so usually the covalent bonds were drawn, for example, when we look at the chemical structure, when you have this carbon and another carbon, and maybe here then oxygen. Oh, no, that's not usually a right way. Carbon, and so this is the reason why carbon, the organic molecule in organic molecule, carbon is the only element that is possible to form this complex macromolecule, because the carbon can have 4 different. Let me put this two-dimensional way. Carbon can form the 4 different directions of a covalent bond, and here some other element can come, and another carbon can come, and even double bond can come, just like that. So by doing this, the carbon can provide this backbone of huge expansion of this covalent bond. That's why carbon is so good at building the complex macromolecules, and that behind that there is a specific type of bond called covalent bond, and that is the most stable. And so the stable bond on also, if you break a single covalent bond, if you break the bond, then energy will be released, and usually very high amount of energy can be released. So it is very difficult to establish a bond, but the other way around is also true, so that if you break that bond, then you will have a bigger reward. It is probably That's what you can, although in actual real reason that is not even a correct statement, but just take it as it is. So that's the reason why this glucose molecule is a very preferred form of a food molecule, because if you break each of these covalent bond, then you will get a relatively very high amount of energy released, is the reason. The second type of a chemical bond is ionic bond. This is also something to do with the electrons. The ionic bonds are only made possible through 2 or 3 different ions. So, for example, sodium element is very easily become ionized to become a plus positive ion after losing 1 electron in itself. And once it becomes this ion instead of this neutral element, and then it can easily have any relationship with the oppositely charged ions, just like chloride. After this chlorine became ionized to be a chloride after losing 1 electrons, and then once they have, and then electrically they are attracted to each other, then they form some specific relationship, the ionic interaction. And this ionic bond is relatively unstable, which means relatively weak, especially if you put any of those elements made through ionic bond. If you put them into the water, then individual water molecule itself is something that charged molecule, so they will try to interfere in their relationship so that they will easily be dissociated. That's why in the water there are so many ionized molecules are dissolved, and we we call those ionized elements as electrolyte or minerals some other times. The third type of a very important chemical bond in life is hydrogen bond. The hydrogen bond is established between one hydrogen, which was bound with other elements through the covalent bond. For example, water. Like I said, in the water there is actually unequal relationship do exist. So here this is slightly negative and this is a slightly positive at the end, because of that reason that this oxygen is very greedy in their electrons. So imagine if there was another oxygen molecule nearby exists, which is the case usually. What would happen is here now between this oxygen and this hydrogen, they can be attracted to each other because of this partial charge, and their distance is close enough. So in reality they have some real interaction, chemical interaction established in between, and this is called the hydrogen bond. So water as a liquid are maintained, and in its chemical property, and liquid water is actually made possible through all the hydrogen bond among individual water molecules are the one responsible for this hydrogen bond. And this hydrogen bond is very weak. Weak chemical bond, so it can be easily broken. It doesn't take any too much energy to break one hydrogen bond between two molecules, but also it's very easy to reform. That's why water molecules is a kind of chemically liquid in property, because it's very constantly some part is building a hydrogen bond, and some at the same time some other part is breaking its relationship. So this hydrogen bond is very weak, but still this is extremely important chemical interaction in biology, in life. In particular, for example, DNA exists. Some of you might have heard that DNA exists in double strand. That's why usually DNA is depicted as like this kind of a helical double stranded, and in between these two strands, the actual chemical nature of this interaction that is making possible for this two strand stick together is the hydrogen bond. Although it is very weak, but it is extremely stable enough to maintain these two DNA strands together. Because although individually it is very weak bond, but it's just like a Velcro, all together it provides very strong enough withholding power. So using this hydrogen bond, many important activities, so it's just like maintaining DNA structure and also enzyme reaction. In biology, in an enzyme, there is a very specific part in any different protein, the enzyme protein, where actual chemical reaction take place. That particular special location of the enzyme we call as an active site. In the active site, very specific hydrogen bond usually. Not only hydrogen bond and some other like ionic bond also takes place, but mainly a lot of different hydrogen bond between the substrate, the target molecule, and the enzyme's active sites are the one actually making possible for this chemical reaction to take place. So anyway, and some other very weak chemical bonds like van der Waals force and hydrophobic interactions, so also do have, but I will not mention those two chemical reaction. The only purpose that I want to bring up these different types of chemical reaction at this point is to introduce you at least the the covalent bond and the hydrogen bond and the difference between these two different types of a chemical bond. And as soon as you can understand these two, and that will be just enough. So types of chemical bonds in biology are actually abound in not only these three, but also hydrophobic interaction and van der Waals forces do also exist, and they also play very significant kind of roles in everyday maintenance of biological activity. But here I will not go over these characteristics. With that, I think before we go into the actual chemical evolution and Darwinian evolution procedure, which will involve the actual real procedure, probably I will have to stop here and then leave the discussion in the next session.